So in our previous two lectures, we completed the glycolysis, and in second lecture we completed TCA cycle as well as the intermediate pathway. In this lecture, we are going to talk about the last part of respiration, without which respiration cannot be completed, that is electron transport pathway or oxidative phosphorylation. But before we go there, I just wish to ask you people that in glycolysis, what was the total ATP yielded? Can I know?
2 ATP.
So ATP produced in glycolysis was 2. And what was the number of NADH produced?
2.
So ultimately these two NADH, just give me a moment. So in NADH, it is also written as NADH plus. 2 of it is produced, which will be multiplied with 3 ATP, and there will be production of 6 ATP for this. So in glycolysis, if I see, there will be production of total 8 ATP. Similarly, if I look into intermediate pathway in which pyruvate is converted into acetyl-CoA, there is production of NADH. So we have to remember one thing, that this intermediate pathway, for conversion of one pyruvic acid to one acetyl-CoA, there will be production of one NADH plus. But we know that from one glucose molecule, 2 pyruvic acids are produced and 2 acetyl-CoA will be produced. So in intermediate pathway, there will be production of 2 NADH, and thus there will be production of 2 multiplied by 6, that is 6 ATP. So up to intermediate pathway, there will be production of 8 plus 6 ATP. How much it is? 14. But we know that in aerobic respiration, there is production of 38 ATP. So 38 minus 14, how much? 24. So these 24 ATPs, production of these 24 ATPs, we have to calculate how and why. Okay. So what are the portions where ATP is produced? One is here. Second is there will be production of here. Apart from that, you know that there is production of FADH2, which will yield 2 ATP, and there will be one production of GTP, which will be in turn once again converted into ATP. So total of these multiplied by 2, that is 12 ATP produced in each TCA cycle. Understood? Then 12 multiplied by 2 will be equal to 24. Why? Because from one glucose molecule to pyruvic acid, that means twice the citric acid cycle will go. But हम अगर देखें for glycolysis TCA cycle and intermediate pathway, we will feel, ma'am, it is ख्याली पुलाव. ये क्या चल रहा है? ATP तो बन नहीं रहा है. We are considering that NADH plus and FADH2 is going to produce ATP. So today we are going to learn how NADH plus and FADH2 is going to get converted into ATP. So this pathway is known as terminal respiration. Why we call it terminal respiration? Because without it, complete oxidation of carbohydrate is not complete. Okay. It is also known as electron transport system or ETS. Why? Because this is the way actually the electrons or high energy electrons are getting transported, and while they are getting transported, indirectly we are using their energy to produce ATP. Okay, and it is also called oxidative phosphorylation. So three of its name are having their own reason. Third name, that is oxidative phosphorylation. Why? Because oxidative, because there will be oxidation reaction obviously, and final electron acceptor will be oxygen, so which gives the name oxidative. And phosphorylation means addition of phosphate. In this process, ADP will be phosphorylated with inorganic phosphate to produce ATP. So definitely we can say it is oxidative phosphorylation. So let's go for the process, how it is done. Actually, I feel like telling you that how efficient organelle mitochondria is. Don't you think mitochondria is a very efficient organelle? Let me tell you why and how. If you look into structure of a mitochondria, different parts are responsible for different types of pathways. Okay. In case of mitochondria, if you see, the inner membrane is folded to form finger-like structures, which is called cristae. All of you know that these inner membranes increase the surface area by having this kind of folding. Now, inside this membrane, there will be presence of matrix. Let me represent matrix by green color. If you look, in oxidative phosphorylation, or before that TCA cycle, which is a very important part, it occurs inside this matrix. So TCA occur inside matrix. Glycolysis occur in cytoplasm. And now we are going to talk about the terminal respiration. It is occurring in the specialized tennis racket-like structure which is present at the inner membrane. So in mitochondria, the gap between two membranes are known as perimitochondrial space. So this gap is known as perimitochondrial space. No, that's why I am mentioning it so specifically. If you see, this is the outer membrane. This is the outer membrane, and the cristae is made by the foldings of inner membrane. I will fill in the area so that it will be easy for you to understand. The area I am highlighting with pink, can you see in the image, is known as the perimitochondrial space. So here, when I am talking about movement of something, or I am talking about proton motive force, we talked about it already in the chapter photosynthesis, where there is movement of proton between the thylakoid and the stroma, isn't it? Here the proton motive force force will be guiding proton from matrix via inner membrane to the perimitochondrial space. Is this part clear? If anyone is having doubt, can ask me question. We need to understand where oxidative phosphorylation takes place. Let me do one thing which will be helpful to you. I will enlarge this part and explain. Consider this is the outer membrane. Okay. And this is my inner membrane. The matrix is present here. And if you see, here there will be presence of tennis racket-like structures which are known as oxysome. Okay, these oxysomes are known as F0, F1 subunit. They are having this stalk-like structure is known as F0 subunit. F1 subunit. And this is F0, the head. And F stands for Fernandez-Moran subunit. So why they are called oxysome? Because they help in oxidative phosphorylation. So they are basically responsible for formation of ATP. So proton is moving from where to where? From matrix to first it will move from matrix to the perimitochondrial space, which is between the outer and inner membrane. And then through this F1, F0 subunit, it will come inside. So I just want to tell you about the oxidative phosphorylation. It is occurring in between the matrix and the perimitochondrial space. Okay. So we will move further to have a look into the diagram. I will first share with you the diagram that is described in your textbook. So we can definitely study from it, but I wish to draw a simple diagram which will help you to understand it better, and we can definitely do that. I will draw, this is the outer membrane. Okay, and this is the inner membrane. So inside this is the matrix. So in the wall of inner membrane, there are a few types of complexes. Why we call them complex? Because they are group of enzymes as well as electron carriers. So first complex that is located is complex 1, then complex 2, complex 3, and complex 4. Apart from that, as I mentioned already, that in the inner world, there will be presence of the oxysome. Okay. So now, how these complexes are helpful that we are going to see. So the complex one, I am writing name separately, is known as NADH dehydrogenase. So in these complex, there is one main enzyme which is called NADH dehydrogenase. And apart from that, a few electron carriers are present. So what are these electron carriers? These electron carriers are FMN and FES. FMN full form is flavin monoadenine nucleus, and FES is iron sulfur center. Basically, all these electron carriers, as we studied in photosynthesis, are having one metal at the center, and then they carry electron by continuously getting oxidized and reduced. But as electron is moving from one electron carrier to the next, the energy of electron is getting reduced. That's why it is always said that the electron transport system is a downhill reaction. मतलब जब वो फर्स्ट कैरियर के पास है, the amount of energy present in electron, in the second carrier the electron will be reduced. Okay. जैसे समझो गरम रोटी को हम हाथ से पास करते, पता है, दो हाथों में करके फाइनली वो ठंडा हो जाता है. It is having similar effect. So okay. So second one is complex 2. Acha, she requested me to write the full form of FAD, so I am writing flavin Acha, you told me to write FMN, na? It is flavin mononucleotide. And FES is the iron sulfur center. So what happens is, in complex 2, the complex 2 is an enzyme system where we have succinyl dehydrogenase. Okay, if I am talking about complex 2, it is having succinyl dehydrogenase complex. Why we call it succinyl dehydrogenase complex? Because one enzyme mainly is leading, which is known as succinyl dehydrogenase. But other than that, electron carriers are also involved in that. Next is complex 3. This is known as cytochrome BC1. So you need to know what do we mean by cytochrome. Cytochromes are metalloproteins, and there are various types of cytochromes involved in electron transport of photosynthesis as well as here. So this is having B and C1 cytochrome. So complex 3 is made up of B, C1. And finally, there is complex 4, which is having cytochrome A, C oxidase. In many books, there is mention of a fifth complex. If anywhere there is mention of fifth complex, that is counted somewhere at the part of cytochrome C oxidase, somewhere it is separated, that is cytochrome AA3. Fifth complex is cytochrome AA3, and it is the final stone in the electron transport pathway. After this, the electron is achieved by the oxygen. So I am writing about the complex number 5, So complex 5 is cytochrome AA3. So it is complex 5 and it is known as cytochrome AA3. Now let us understand the concept of electron transport. When we talk about electron transport, we know that by TCA cycle, by glycolysis and by intermediate pathway, continuously there will be production of NADH H plus. If you can remember, I told you that one molecule NADH plus H plus is having 2 protons, 2 electrons and 1 NAD plus. Okay, this is already discussed in photosynthesis, and also I am discussing it here. Similarly, what will happen when NADH will come to complex 1, it will dissociate. That means the NADH will dissociate to produce NAD. So what about the protons and electrons present in them? I request everyone to pay attention. This lecture is anyways getting recorded. So the protons will be transported outside. Okay. And who will give energy for transportation of these protons? The energy of electron is used to transport proton. Understood? Is this part clear to everyone? In each molecule of NADH plus, there will be transport of proton. When there will be dissociation of it, there will be release of proton. That proton will be transported from matrix to the perimitochondrial space. Who is giving this energy for transportation? Energy of electron. So the way proton is going outside, for electron, we have to understand electron will be donated to first FMN, then it will be donated to FES, and then it will be going to Q. What is this Q? Q is quinone. So I am just writing about Q. Q is known as quinone. The full form of Q is quinone. It is also a kind of electron transporting system. Okay. Now, similarly, when I am talking about FADH2, FADH2 cannot be available to first complex. Why? What is the name of first complex? NADH dehydrogenase. So it is having ability to dehydrogenation reaction of NADH, not FADH. So obviously in this case, it will be possible So we were talking about FADH2. In this case, the FADH will be coming to complex 2. So for this purpose, FADH2 will be releasing the proton. Same way, the electron released by FADH2 also will ensure the transportation of proton in the perimitochondrial space. Now, the electron carried by this FES will be further carried by Q, and also the electron that is released by FADH2 is also carried by Q, and this electron will come to complex number 3. So if you look into the structure of complex number 1, 2, 3, 4, and obviously if we talk about complex number 5, NADH is able to use, so for NADH plus H plus, it will go to complex number 1, 3, and 4. The proton derived by NADH H plus will pass through complex number 1, 3, and 4. Whereas the electron derived by FADH2 will go to complex number 2, 3, and 4. That means complex number 1 can never be used by FADH2 to send proton outside. And similarly, complex number 2 can never be used by NADH to send proton outside. Okay. So, same way, further once again the protons will go out via complex 3 and complex 4 is responsible to mainly for electron transport, and from Q the electrons will be transported to complex 3 and then to complex 4 and then to complex 5, and finally the electron will be accepted by half O2, that is oxygen, to give rise to O2. Okay. So this is the way the final electron acceptor is O2. Now you must be thinking कि मैम ये सब तो समझ में आया, अभी तक अपना साइटो ऑक्सीजन तो यूज हुआ ही नहीं. So as by using the electrons energy, protons are moving outside into this place which is known as perimitochondrial the proton present outside is more as compared with the proton present inside the matrix. Isn't it? Why? Because as soon as proton are carried by NADH and FADH2, they will be transported outside. Via whom? Via these complexes. So what will happen? The proton present in the perimitochondrial space will be obviously more than the proton present inside. So there will be development of a proton gradient. What will happen? There will be development proton gradient. What do we mean by gradient? Rutu, can I know what is gradient? Ayushi?
Difference.
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 So outside the charge, that is positive charge, will be more. Outside where? Perimitochondrial space. And inside, in the matrix, the proton number will be low. so it will be less positively charged. Now using this energy, the proton will be transported from outside to inside. And as I discussed in photosynthesis, that this head of oxysome is having ATPase. What it has? It has ATPase enzyme. So as soon as proton is pushed inside, there will be presence of whom? There will be presence of proton motive force. And by using the proton motive force, ADP will be converted into ATP. So who is doing this? It is done by proton motive force. So if you see in ETS, that is electron transport pathway, very important things are occurring. First thing that is occurring is electron flow. So in ETS, three main incidents are occurring. One is flow of electron. So where is flow of electron? Electron की flow कहाँ होती है? Through the electron carrier. And as it is moving from one carrier to the next one, it will lose energy. ठीक है? So one incident that is occurring is flow of electron and energy release. So this energy is used for what? This energy
Protons
is used
to move
for
outside.
so it will send proton from matrix to perimitochondrial space. Okay. Second incident, which is equally important, there is proton movement and gradient. So the gradient is developed due to outward movement of proton using energy of electron, and then proton will come back into the matrix, and by using that proton motive force, there will be phosphorylation by ATP. Third very important thing that occurs is the proton motive force driven ATP production. Because ultimately the main aim of this terminal respiration is to produce the ATP. Okay, so this is all about the electron transport system. Now I wish to answer one question of yours, that why in case of NADH plus there is production of 3 ATP, whereas in case of FADH plus it is that, or FADH2, there will be production of 2 ATP. Actually it is not 3 and 2, it is actually 2.5 and 1.5. I'll tell you the calculation. Through complex one, I just will use another page. I guess that this part is clear to everyone. Anyone have any doubt regarding this part? Feel free. Yes, beta, bolo.
मैम मतलब दिस ऑक्सीजोम मैम वो मतलब वो जो प्रोटॉन ग्रेडियंट जो क्रिएट हुआ है उसको ओवरकम करने के लिए प्रोटॉन्स अंदर मैट्रिक्स में पुश करेगा?
नहीं, वो पुश नहीं करेगा, उसके थ्रू मैट्रिक्स में वो बाय डिफॉल्ट आएगा, बिकॉज इट इज अ टेंडेंसी ऑफ एवरीवन टू मेक इक्विलिब्रियम, and the inner membrane of mitochondria is impermeable due to its hydrophobic nature, so it doesn't allow proton to move. Only space by which proton can come inside is the F0 subunit and F1 subunit. Understood? वो कुछ नहीं करेगा, वो बस प्रोटॉन के मूवमेंट को चैनलाइज करेगा. F0 will act as a porine protein which will channelize the flow of proton, and F1 by using that energy which has ATPase will synthesize ATP.
Okay.
So in first case, the transport of proton from matrix to perimitochondrial space is by active transport. Why? Because it is using the energy of electron, isn't it? But when, and it is against concentration gradient. But when I'm talking about its movement is from perimitochondrial space to matrix, this is by passive transport because it is towards the concentration gradient. Is it clear?
Yes, ma'am.
Any other questions, anyone? So we will talk about the next important part. I'll make you know that how NADH is producing free. Actually, you know that in case of NADH plus H plus, there will be production of like it is passing through complex 1, complex 3 and complex 4. So during movement, number of proton passed in case of complex 4 is, so 4 electrons are 4 protons are passed through complex 1, 4 protons are passed through complex 4, and 2 protons are passed through complex 4. This is the number of proton at a time it can pass. Okay. Whereas in case of FADH2, the number of proton that is passing from complex 2, complex 3 and complex 4. So it can pass 2 proton each from complex to complex and by F0 F1 subunit, every time during transport 4 protons are transported. So here it will be 10 proton and here it will be 6 proton. So it will go outside, this will go outside and this will go inside. So if I calculate in case of NADH plus 10 divided by 4, what is the value we get? It is 2.5. Why 10 divided by 4? Because by for movement of 10 proton outside in the perimitochondrial space, 4 protons are coming inside and 4 protons are required to produce 1 molecule of ATP. Understood? So 2.5 ATP is produced, and in this case it is 6 divided by 4, which is equal to 1.5 ATP. But in NCERT it is rounded off to make it 3 ATP, and it is rounded off to make 2 ATP. So this is the calculation. Okay, so that's all about electron transport system. One short thing I just wish to say, all of you must have heard about cyanide poison. सब बोलते हैं कि उसका टेस्ट कैसा है, कोई लिख के नहीं जा पाया क्योंकि everyone wrote S. It's not known that it is salty, sweet, sour, what it is. Because S लिखने के बाद they die. Actually, cytochrome attack. Cyanide attack cytochrome A8. which will finally donate electron to oxygen. And due to invasion of cyanide, the oxygen cannot take electron and the oxidative phosphorylation remains inactive. And that's why a person have instant death without presence of ATP. It doesn't allow. जो इलेक्ट्रॉन मूव हो रहा है उसको फाइनली किसी को लेना तो पड़ेगा ना, and the final electron acceptor here is oxygen. It doesn't allow that oxygen to receive electron, and that is why it is a killer and deadly poison. So I already shared the PPT with you people, and the details are mentioned over here, but it's not present neither in NCERT nor in this such detail. And so we can understand that why the complex, that is 4 of the main complex and cytochrome AA3 is responsible for production of ATP. Now we will quickly move ahead and I want you people to make a balance sheet of respiration. As I said that up after the end, you should be having clear idea that how many of them are produced in TCA cycle, glycolysis and others. Okay, so these pathways are working in simultaneously. ऐसा नहीं है कि जब तक सारे ग्लूकोज में से TCA cycle हो जाए तब तक ETS बैठा रहता है, because that is not possible. All of them are working simultaneously, and it is on demand that whatever respiratory substrate available, they can be controlled simultaneously. So this is the balance sheet, and I wish all of you to look into it, and you will be able to understand how 38 ATPs are formed. We discussed it repeatedly. So out of these 38 ATP, 2 are used, and these 2 ATPs are used for transporting the substances from cytoplasm to the mitochondria, and that's why the net gain is 36 ATP molecules, but production is 38. So this is the comparison between fermentation and aerobic respiration. You can see that from the point of view of energy gain, aerobic respiration is far more superior, but it doesn't give delicious product like lactic acid or alcohol. So that is the disadvantage. Now we will talk about the last topic of this pathway, that is amphibolic pathway. It is called amphibolic because in this both the catabolic and anabolism takes place, and it is about utilization of respiratory substrate other than carbohydrate. At the beginning of this chapter, we talked that the glucose is not the only respiratory substrate. ऐसा नहीं है कि हम सिर्फ ग्लूकोज ही पीते रहते हैं, हम खाने में तो कुछ भी खाते हैं. So how then others are responsible to be utilized for TCA cycle? Glucose is the most preferred one, but in its absence, other respiratory substrates can be used. I want everyone to pay attention here. See, fats are first digested to form fatty acid and glycerol, and then they can come to respiratory pathway, that is the TCA cycle, by 2 methods. One, some fatty acids and glycerols can be converted into dihydroxyacetone phosphate, and from that they can go for rest of the glycolysis and then TCA cycle, or they can directly be converted into acetyl-CoA and utilized in TCA cycle. So 2 options are available for fat. In case of protein, obviously after digestion they will be converted into amino acid. These amino acids can be converted into pyruvic acid and can be utilized in PCA cycle. So this is how fat and proteins can invade in between TCA cycle and can be utilized. Okay, and this pathway is known as amphibolic pathway. Now I wish to talk about RQ. We already talked about PQ, that is photosynthetic quotient. So photosynthetic quotient PQ is equal to volume of O2 released divided by volume of carbon dioxide absorbed. Because in photosynthesis, the order is different. Photosynthesis, we plants absorb CO2 and release O2. Here in case of RQ, it is volume of CO2 evolved divided by volume of O2 consumed. So basically, it is a ratio of CO2 evolved and O2 consumed. So if you see for a carbohydrate, the RQ is 1, because if you see the equation of respiration, 6 O2 is consumed to give 6 CO2. So 6 divided by 6, the ratio will be 1. But for fat, it is less than 1 or equal to. Why? Because in case of fat, actually it takes a lot of CO2 release to with respect to O2 consumed. And for protein, it is 0.9. So by RQ, you can understand that you can identify by looking into the RQ that respiratory substrate is protein, lipid or carbohydrate. Okay, so that's all about this chapter, and with this we are done with the chapter number 14, and we are done also with the syllabus of grade 11. Okay, so big congratulations to all grade 11 students. But I said that the syllabus is finished by me, not by you. You need to practice these pathways very, very, very efficiently. So the forum is now open for discussion of doubts.